So many of you don't know that. And one of the few people that have been around at the Institute for that long, right, is your next speaker. His name is Dr. Vadim Jucaud. You all know him. I've been working with Dr. Terasaki for quite some time as well, but on a day-to-day basis, I was just kind of doing the math. And I think Vadim is really the one that has been doing, been here the longest and working directly with Dr. Terasaki. I mean, if you count the time that he was at the laboratory, about 16 years. So yeah, I worked with Dr. Terasaki throughout the years, mainly in UCLA. I was with him for about 5 years. And then once we started and spun out the company One Lambda in 1984, I didn't see him as much. I saw him, of course, at the board meetings and things like that. And we would have periodic meetings in between. But like I said, I think, direct day-to-day working relationship, I think I have to hand that crown over to Vadim. Anyway, so super happy to be able to have this talk today. Johnson and Vadim and I started talking about this about a month ago, just different kinds of ideas to be able to commemorate Dr. Terasaki's birthday. And so we're super happy that Vadim has offered to talk a lot about the legacy of Paul Terasaki and his original accomplishments. I think many of you, you might have a nice idea of what he's done, but I think Vadim will go into a little bit more detail about his actual accomplishments and where he made impact in the Institute. And that's really, what the Institute is doing today and trying to continue Dr. Terasaki's legacy, right, is making impact in the clinic. So without further ado, I'd like to let Dr. Vadim Jucaud come up to the podium.
Good afternoon, everyone. Thank you very much, Stuart, for this kind introduction. So first, I really want to say that I'm really honored to do this presentation for you guys because, I've been here for quite some time now, more than 15 years. And again, I started my research career under the mentorship of Dr. Terasaki. And I think this was one of the best opportunity I had in my life. And, I had really the luck to work with him. So today, really, I want to, do a tribute to his life, his legacy, and his global impact, and really trying to describe how he changed the life of many people, including myself. And we're going to do that by going through, all his life journey as a pioneer in transplantation. And I'm going to try to, give you guys a perspective of the next generation of scientists in the field of transplantation, which was dear to Dr. Terasaki. And I think, I'm somewhat of an example of him trying to, continue the research in the field that he started. So without any further ado, let's start again by this, I'm going to really showcase to you really the life of this transplant pioneer from the beginning until today, because I think his vision is still alive with us today at the Institute. And again, I think his impact globally, it was not only by saving lives through his science, but also, again, like I said, he inspired, the next generation of scientists and people, with his generosity and his humility. So, for me, Dr. Terasaki was one of the brightest person I've ever met. And I think, again, his best quality was his humility, where, even though he was a legend in the field of transplantation, he was always open to discuss with anyone and share his knowledge and experience with people. And I think this, really what made him a great scientist. So with that, Dr. Terasaki, he was born in 1929 on September 10th. So today would have been his 96th birthday. So he was born in Boyle Heights in Los Angeles. So again, he's Angelino, which, I think this is important to still mention because, he spent all his life and career in Los Angeles. And I think, even myself, even though I'm from France originally, I was born in LA. And I still take pride in, being an Angelino because this is where really great thing happens. And so here, this is a picture. So not back in 1929, but the house in Boyle Heights with his father in front where he was actually born. And so Paul Terasaki had also a Japanese name, Ichiro, which actually he was using until he went to college. And he decided to change his name to Paul because he thought that many people didn't know how to pronounce his Japanese name. So I think this is an interesting story because I always knew him as Paul Ichiro Terasaki, but by preparing this talk, I realized that actually, his name is Ichiro, not really Paul. So I thought it was pretty interesting. So here you see a picture of him with his father at 2 weeks old in 1929. And so Dr. Terasaki was the firstborn child of three brothers, and Ichiro really means firstborn in the Japanese. So again, Dr. Terasaki, I think, really kept his culture, the Japanese culture, really deep anchored in his heart. And I think even myself, working with him, I learned so much about the Japanese culture and how they treat people with respect and integrity. And I think this is also an important point to mention because I think what he did throughout his career was always with respect and integrity. So here you see him on the right side of the screen, at the beach with his mom and his little brother, and he was about 2 years old there. So then the next part during his youth, so he stayed in Los Angeles until 1942, until the war started in the US. But before that, I wanted to show this picture to share that, I think Dr. Terasaki here was part of the Boy Scout troop. But, this I think shows that, he really cared about also community and being part of, I think a bigger picture. And I feel, then again, they never told me that, but I feel that this is a good example of where his sense of, community and, doing good among groups of people probably started. And one thing that was interesting that he said in his scrapbooks is that really he was the youngest and the skinniest guy over there, but this doesn't prevent him from actually joining this Boy Scout. And this you'll see later, where I think is really, something that probably shaped him in terms of like his sense of community and helping others. So then in 1942, with the war, him and his family went to the Gila River internment camp in Arizona, where he lived there until 1945, when after the war and when he was released. And again, I think, in this camp, he, also probably shaped a lot of the way he viewed life, because in those internment camps, life was not necessarily easy. Resources were kind of scarce. And I think also later on, you'll see how, understanding how to manage resources was probably very important for him and some of the discoveries and impact that he had in science. But one thing that was important, again, linking back to the Boy Scout, even in this internment camp, he was still part of a Boy Scout troop, which again, to me, also demonstrates, his sense of community and really being part of, a bigger, but again, good community. And I think he always knew that, you can do great things on your own, but he was really, I think, a team player and understood how, in a team you can go further than on your own. So then after the internment camp in 1945, he actually moved to Chicago for a little bit where he started college. But then from Chicago, he actually came back to Los Angeles and transferred to UCLA. And so this is at that time in 1948 where he became a Bruin. And to me, what's very important with that is first, he was accepted as a transfer student at UCLA, which, is, for transfer student is, sometimes a little hard to get integrated in the university that way. And I say that because I was myself a transfer student at UCLA. And I think those type of things also, helped me connect a little bit with him. and understand that even, you're maybe a transfer student, you can still achieve great things later on in life. One thing that is very important, or I think that I want to mention, which he also mentioned, is back then UCLA was $39 tuition for a semester, which I think, was great, given that for him was probably a really great investment for his education and his future at that price. And then after, during his time at UCLA, I think he mentioned again in his scrapbook two people that were really instrumental in him going to research and really performing the research at the level that he did. So the first one was one of his lab mate during graduate school, Mary Millard Cohen, which really taught him all his technical skills in the lab. And I think, this is one of the things that he always told me when I was in the lab, because we were doing plenty of, clinical research where the technical skills is the foundation of the science, and you have to keep that at the highest level to be able to get the best result that you want to get. But again, when you have this really high rigor in the technical skills, then definitely you can reproduce experiment, and people definitely will trust what you're showing. So I think that lab mate of his really, helped him, understand and realize that really any research start with rigor in the lab experimentation. And then the next person is Dr. Abraham Shetman, who was his advisor during his PhD thesis. And what he mentioned that he taught him is really the love of how to perform science and how to really perform science in an effective way. And this, I think, you would realize later how this was important in him making those fundamental discoveries, because I think he understood that from an observation to describe the outcome of an observation, it could be very simple. And the way is maybe you have to look at the outcome rather than start at the beginning and trying to get to your outcome. Because in a sense, as if you are able to with an experiment, show that the outcome as what you think, then you can let others figure out all the steps to get to that outcome. And again, I think that outcome is really driven at that time towards patient, and the center of the research was really the patient, because at the end, Dr. Terasaki was really motivated to have an impact on patients and helping saving people's lives. So one thing that was interesting during his studies, he actually in 1954 got married to his wife, Isako. So I think, I myself during my studies, I guess when I was doing my PhD also, got married. And so I know the struggle or the, hurdles that this can take. And I think, it's magical on one side, but also can be a little hard, but still, I think he had love of science and his wife where he was able to achieve that. And then from that union came their first son, Mark Terasaki. And again, I relate to that experience a lot, because also myself, I had my first or my daughter when I was writing my PhD thesis. And I think it's it's difficult, but still with the love and passion for both research and your family, you can achieve about anything. And I think this picture is a nice picture, because you can see him still reading newspaper, being intellectual to some extent, but still keeping his family close. And I think with everyone that knew Dr. Terasaki, he was a family man that always kept his family close all the time. So yeah, so then after that, in 1956, Dr. Terasaki earned his PhD from UCLA, and back then the department was called the zoology, which I thought is very interesting. When myself at UCLA, the department was called EEB, which is evolutionary ecology and biology, which the very first I thought I was doing biology overall, but now they have a very complicated name. But back then, biology was called zoology. So I thought, this is very, interesting how diploma naming evolved. But in this picture, what I think was, very important for him probably, and which he mentioned, is that, his parents were very happy for him to graduate. But one of the reasons is because him coming from a family of Japanese immigrant, I think he was the pride of his parents to, be a generation to have a doctoral degree coming from a family of Japanese immigrant, which, from the earlier times was not all that easy. And also another important or, funny fact at that time is actually on the same day, his younger brother graduated with a bachelor's too from UCLA. So I think, their parents were doubly happy about the achievement that their kids were able to do. And I think, so from there in his study, Dr. Terasaki, I think this is where he really, established all his techniques and also his love for organ transplantation. And his early work really started with chick embryos, where he was doing cross-transfusion experiments to sort of understand how blood transfusion impact the survival of chick embryos. So again, at that time, in 1956, there was not much understanding of how the matching or how transplantation happened, because transplantation, as a reminder, is a very new field of research. The first transplants were done in the 50s, and so comparing to other diseases, transplantation is really a new field. So at that time, you really had to have a lot of imagination and creativity to actually perform the experiment. And I think this experiment on chick embryos, was really what pushed him to in the next step in his career, where many people throughout the world were really impressed with his work and invited him to be part of different labs to perform some postdoctoral study. But one thing that is very anecdote about that is Dr. Terasaki worked with so many chick embryos that later on in his life he never really ate chicken. And so this, if you invited him for dinner, then you had to make sure that you didn't cook any chicken, and this really, I think, it started from all those experiments. So this also showing the hard work that he was always doing, because to not want to eat chicken, he probably has handled thousands and thousands of chick embryo. So again, they're showcasing his dedication really to research. So then after he did all those experiments and got his PhD, he wanted to apply to a postdoctoral position at Sir Peter Medawar lab in the UK. And Sir Peter Medawar eventually went on to get the Nobel Prize. And but at that time, he really was one of the major player in the budding field of transplantation. But the interesting story is that he first got a rejection from Sir Peter Medawar. And what he says is that he was very disappointed when he received a courteous reply stating that he had no space for him. But what he managed to do is that he met another colleague of his called Leslie Brandt at Caltech, and he knew Medawar, and so his friend Leslie Brandt really was able to contact Medawar and convince him that the work of Terasaki was very important and that he should take him in as a postdoctoral student. And eventually, Medawar called Terasaki back and said, "Yes, now we do have a space for you because I think your work is great." And I think this is an important point, and especially for me, is that this shows that connections and people sometimes help in shaping where you're going next. And you'll understand in the next slide why I'm saying that, because I think there's an interesting back end story of some of the reason why I'm here as well. But again, so then in 1957, he moved to Peter Medawar lab, where he continues his work on the cross transfusion of chick embryo and really understanding how blood components had an impact on the survival of those chick embryos. So after really understanding how antibodies within the blood as well as immune cells within the blood both impacted the rejection or tolerance of those transfusion in those chick embryos. So after 1 year at the Medawar lab, he wanted to actually go to the Professor Jean Doss's lab in France, who eventually he became the Nobel Prize winner for the discovery of HLA. So he actually applied, had a meeting, with Jean Dossé at his lab in France. But one thing interesting again is like when Jean Dossé discovered that Dr. T's French was limited, he sort of changed his mind and did not really, welcoming with open arms into his lab. And then I think, again, what I think is very interesting in this part is maybe one of the reasons why I came here. I had during my undergrad studies, I did an internship in France in the lab where the lab director was actually students of Jean Dossé. And so I think it's interesting how, a student of Jean Dossé recommended me to Terasaki. But then, I was lucky enough that Terasaki, I guess I spoke good enough English that he, took me into his lab. But so, again, with that, even though he got a rejection from this lab, and again, this to show that rejection is okay. I know all of us as scientists, when we submit papers and things like that, we're always afraid of rejection. But still, I think from that rejection, he still thought that he had a lot of, research ideas that he really wanted to investigate. And he made the decision to go home because he felt that that's where he could actually do it. And home, again, was Los Angeles. And I think, from that experience is really when he made his one of his biggest, or probably the biggest discovery that he made. And I think it's from, again, the challenge of rejection, still going back, having plenty of ideas and working very hard towards, that one goal that he had. And so in 1964, that's where he introduced the microcytotoxicity test. And so what that test is really a test to detect antibodies. Before, the way to detect antibodies was really using a large volume of blood, was not very specific, was not very scalable. And then because of that, yeah, he thought that he had to do something about it. And I think using a little bit of technology, he was able to create this microcytotoxicity test where he was using this little tray that you see in his hand to create a test where you can do, add recipient and blood sera, and you can do that with only 1 microliter of blood to do the test. And I think, this really is why some of the technology really got adopted by the whole transplant community overall. And again, like one of his quotes, what he was saying is that, the first trays that they made, they used a cover glass with an aluminum side, and it was using also some wax tray. So really crude method, but he submitted that in Nature in 1964, and really, it became, as with his own word, it became a classic, drawing thousands of citations. And again, the impact of this test is immense for the transplant community, and really, if transplantation is nowadays possible, is in part due to this test. And I think one thing important to say is, in transplantation, it's a lot about surgeons and how to implant the organ. But Dr. Terasaki was not a surgeon, but as a scientist, he was really able to impact, I would say, even more lives with this test than what surgeon did. Of course, you need a surgeon to transplant the organ, but the surgeon can only transplant so many people in their lifetime. But Terasaki, with this test, again, impacted millions of lives since 1964. And so with that, I want to share here a little video. So of course, of Dr. Terasaki. Yeah. Oh.
Most important thing I did. I often wonder, why is it that, I was maybe 35 years old. Why is it that I couldn't do anything better after that? I had, had another 40 years, and all during that time I worked pretty hard. And how come I couldn't do anything better than that? Maybe it's true in a lot of people that when they're young, they're able to do the thing that they maybe can't do when they're older. The 5 years before that, I think I really worked the hardest in my life. And that was after coming back from England and trying to develop the test. Working those 18-hour days, I was finally able to make the micro test, allowed the development of the HLA field. helping of transplant patients, standardizing HLA. For example, we standardized by sharing cells throughout the world. That couldn't be done without the microtest. So even though the microtest is today replaced by DNA technology, it was critical for the early development of this field. And we couldn't have, in one step, jumped to the DNA, even if the knowledge was there. So I feel that maybe that was this I did. Well, I'm happy that I had the chance to do that.
So again, I think that, I wanted to share this video for, a few reasons. I think the first one, definitely, you can see that, again, he's a very humble person, because I think he says that this was the best thing he ever did. But I hope I'll showcase to you guys that he had still tremendous impact after the discovery or the development of this test. But again, what he mentioned too is that, it's really with this test that he was able to develop the field of HLA, so the human leukocyte antigen, which now, we know that it's, the main element of the immune system. And so really what is that, innovation in this microcytotoxicity test is really the first one is to use 1 microliter of samples, so being able to save samples. And so here, remember, this was a test that was designed for patients. And so when you want to take blood from patients, sometimes the taking sample is not that easy, or the amount of sample is low. After also, when you think of transplantation, many of the donors are cadaver donors, and getting high volumes of blood from those people is also extremely hard. So if you were able to minimize the volume that you used to test, to be able to do the matching between a donor and a recipient, then you could actually do much more tests with the same amount of blood. And I think, again, here we're talking 1964, and this was already a micro test. And, like, even if you want to compare to nowadays, specific molecular tests where we try to detect antibodies like ELISA's, volumes are 50 to 100-fold higher than what he used to use in 1964. Then the next aspect is really that this test really allowed a rapid and accurate and then reproducible result, which, again, in the case of transplantation, some of those tests have to be done really rapidly, because you don't want to waste time to transplant a person, because of course the quality of the donor organs are impacted by how long they stay out of the body. And I think the last point, which is, I think, highly under-recognized, his test was probably, the first example of a personalized medicine assay. And again, I want to remind you guys, this is 1964. I know nowadays, personalized medicine, particularly at our institute, sometimes we take it, for granted that we all know that that's what it is. But I would argue that really Dr. Terasaki was the first person to create a test for personalized medicine. And in that case, here, why is it personalized medicine? Is each transplant is a unique case of a donor and a recipient. So for that, again, there is no, standard way to really do it. But every case is different, but still there, transplantation is personalized medicine. And I think with the, endeavors or the division of the Institute nowadays, we're really not very far from that at all. Or I think Dr. Terasaki was really, ahead of his time in 1964, because we're still, doing that, 50 years later. And so again, after just talking a little bit about the science, what is this test really is incubating together PBMC or lymphocytes or immune cells with the serum that may contain antibody against that cells. And here what we're trying to see is really how the antibodies are able to kill the cells. And so if the antibody kills the cells, it means that the patient has a specific marker on the surface, which later on they figure out it was HLA, and that the sample has a specific antibody that binds to the HLA. So because of that, really, I think, from this microcytotoxicity test really started the first international transplant meeting in Paris. And so why this really started is because with this test, he was really also able to do the HLA typing. So really understanding what HLA gene people, donors or recipient may have. So from that, he was able to, again, do the matching of people and really showing that if you care about matching, then you may be able to improve your survival, which eventually was shown to be true. And then also in this meeting, one important report or presentation that was made by Dr. Terasaki was the presentation of 7 cases of hyperacute rejection. And so in transplantation, there is many different types of rejection, but hyperacute is one of the worst one because it's the one that happens right after transplant that can actually kill an organ from hours to even minutes sometimes if the immune response is really bad. And this is going to be very important for his next seminal discovery, which again, he was using the microcytotoxicity test, not this time to do the HLA typing, but this time to detect HLA antibodies. But before he did that discovery, he also in 1968 had a meeting with the World Health Organization in New York with all the pioneers and all the best people in transplant, because at that time, HLA did not have a name, and really, Dr. Terasaki was the one that named HLA. After how it came about is Jean Dosset, who was the Nobel Prize winner for the discovery of HLA, wanted to call it a human antigen. But then another transplant pioneer wanted to call it a leukocyte antigen because this is where they were able to detect the antigen in the first place. And Dr. Terasaki had the great idea to satisfy both parties and say, let's call it HLA to have the human side and the leukocyte antigen name. And so this is really how HLA naming came about. One thing that I always found interesting in this, and you read that everywhere, even in the scrapbook. So the person that named the HLA antigen, which is Dr. Terasaki, we always say HLA antigen, but in HLA, the A stands for antigen. So it's sort of like we're saying human leukocyte antigen. I always thought that was funny. So I don't know if really the nomenclature is the best one yet, but regardless, we're still the one that is used today. So I think, this again, it's also demonstrating some of the impact he had for the transplant community, because later on we understood that the HLA gene is the set of gene that is the most polymorphic in the human genome, meaning that there are more than like 15,000 different alleles for those genes. So definitely, without a good nomenclature, it would be very hard to really perform research and standardize all the assignments of different genes for different people. Because again, remember that HLA matching is very important for the survival of a transplanted organ. And so after that, again, in 1969, this is where he introduced the Crossmatch assay. So again, here I think it's a good example of him developing a technology in 1964, the microcytotoxicity test, that originally was made to again, detect antibody, but if you can detect antibody, then you can do the HLA typing. So again, to try to see what is the specific HLA type of a person to better match patients. But what he did here is really using the technology for a different type of application. And again, he knew that he was able to detect antibodies, and he was wondering, if you remember again about the case of the hyperacute rejection, where the patients were losing their graft hours or minutes after the transplant. He was wondering why, and he always had in the back of his mind antibodies are the culprit. And in this paper, this is where he tests the serum of transplant recipient before they were transplanted, and with this test realized that they had a lot of antibodies against the donor that they received. And so because of that, they went into having hyperacute rejection. So from this paper, really, he demonstrated that having antibodies against the graft before transplantation should be a contraindication for transplantation. And again, from the paper, if I read the last conclusion sentence, the presence of preformed cytotoxic antibody against the donor appears to be a strong contraindication to transplantation. And because of that, this test really revolutionized transplant matching on the first part, but now revolutionized safety of transplantation, because now we had a way to detect whether a patient would really lose their graft within hours of the transplant, which is something nobody wanted because, again, you lose the graft, you may lose the patient at the same time. An interesting anecdote is that this test was also called the NIH test because, again, it was approved by the NIH because it was a clinical test that at the end was used to indicate to clinicians whether they have to perform a transplant or not. And again, here, I just want to show you just a schematic of what this test is at the end, again, you take recipient serum that should include donor-specific antibodies or not, and then you mix that with donor lymphocytes. And then if you see negative cross-match is when you don't see any killing of the cells because there is no antibody that is able to kill the cells. And a positive one is when you see killing. And again, why this is an important assay is that not only you detected antibodies, but also you really detected the effector function of the antibody, which is killing the graft. So again, this is really a functional assay, not to just say if the antibody is there, but can the antibody have the ability to kill the donor cells. And again, this test, cross-match still remains the gold standard prior to transplant surgery. And still nowadays, if you have to perform this assay right before transplantation. If this is positive, you're not going to transplant. And centers that now have cases of hyperacute rejection would really close in those days standards. So that's why now hyperacute rejection is a type of rejection that is almost gone from transplantation. And again, this is due to this seminal discovery of Dr. Terasaki in 1969. And I think this year, following that paper was big year for Dr. Terasaki because this was the year where he became a full professor of surgery. And he said that this was a distinct honor since he was not a surgeon or an MD, but a professor with limited teaching duties. And, I think this is, very interesting. And again, relating to the fact that transplantation is most of the people that get the limelight used to be the surgeons. But here people really realize that even the work in the lab from a non-surgeon really had a tremendous impact on patients' life. So through his research, he was actually able to save people's life, which is technically what also surgeon, are doing. So I think, it's a well-deserved title for him. But really, I think what is important for me there, is that his from the person that support him, Dr. Longmire, really realized that typing patient before transplantation was really essential for the success of transplant. And here you have an image of, again, a full professor of surgery actually in the lab doing the experiments, reading the results. But on the right side, you can see the amount of tests that he had to perform. So really, because of that, definitely, if he was a professor with teaching duties, probably he was not able to do his research. And I think this is important, especially for us here at the Terasaki Institute, where we have, faculties that are, professors without also teaching duties. So again, I think, his model of research and being still a professor without having to teach in the academical sense was, I think it's a important, identity that we still keep nowadays. And after, I would say also, even though we don't have teaching duties, regular one, I believe that we're still teaching the new generation of scientists on how to perform the right science. So despite the fact that he says that he really had no teaching duties, I think he still taught a lot of things, especially for the next generation of scientists. So then after another really important discovery or impact that he had on transplantation was the development of the cold storage solution. So again, also in 1969 and was adopted in 1970. So again, he created with the collaborator of him, GM Collins, the first commercial organ preservation solution that really allowed the transportation of organ. And so again, allowing the transplantation of more patients further away from where the donor was. And so again, I think you understand how this really impacted the transplantation nowadays, and especially that, again, now we try to improve the preservation of organ because of the organ shortage and being able to keep organ for a longer time to transplant more people. And everything really started from this type of research that Dr. Terasaki performed. So then again, he always focused on HLA research and transplantation. I think his next big milestone was the automation and standardization of HLA typing. And why is this important is because this really allowed the global collaboration between transplants lab to really improve the survival and quality of life of the patient. After also this, what really helped to do as well is to design the organ allocation schemes. And I think with his technology of being able to type patient, he was the one that really started allocation scheme here in Los Angeles to be able to understand who would be a better recipient for a specific organ. And nowadays, all those allocation schemes are still used like worldwide. Of course, they're a little bit more elaborated than back then, but still, again, allocation started at that time where he was able to actually do the typing in an automated and standardized manner. So then after from all this discovery and technological discovery, this I think is the next, big staple or big thing that he did is starting One Lambda, which was a biotech that really focused on transplant diagnostics. And this, one thing very interesting is he developed that microcytotoxicity test in 1964. But you can see that it's really, 20 years later that he founded his company based on his patent on that microcytotoxicity test. And those here are the picture of like the original employees of One Lambda. And I don't know if you can see it or recognize, but our president, Stuart Hahn, was part of one of those original employees there. So, and again, this company, ended up becoming a huge company with more than 350 employees and later on acquired by Thermo Fisher in 2012. And again, in terms of transplant diagnostic test, this was the leading company worldwide, and everyone, trusted Dr. Terasaki, because of his integrity and knowledge in the field. And again, that's why I think, his company did really, really well and also was able to advance the field of transplantation. So then the other impact that he really had, and this he started in 1985, he started a book series. And this book series, from his perspective, was a unique, a resource book for transplantation, where you would really be able to track the entire development of the field. But like I said, this is a really, an early field of research that is not that old. And one thing that he was really instrumental in doing is organizing and being able to create registries and trying to standardize the way people did transplant, but also sharing all the knowledge of the different experience of different centers. And this was a very good platform to do so. After, from a personal note as well, and I think few of people in the audience also, the first paper that Dr. Terasaki always told us to write in was in that book. And because of course, we were publishing it, but, as young scientists, we had to have some training. And of course, always aiming to publish in Nature. It's good, but, first we need to start learning how to write. And then this was a really great platform for us and for Dr. Terasaki to teach us the writing of scientific papers. So then the next really big achievement of Dr. Terasaki is the Medawar Prize. So the Medawar Prize, again, if you remember, was named after one of his mentor, Sir Peter Medawar, and it's recognized as the world's highest dedicated award for the most outstanding contribution in the field of transplantation. And this is really given for only the people that had really a tremendous impact in that field. So, an interesting thing is if you want to see that medal, it's actually right here on the side wall over there for you to admire. But again, this, the only higher prize you can get after this one is probably the Nobel Prize. So again, I think, his impact in the field of transplantation was really recognized with that prize. So then from there, in 1999, Dr. Terasaki retired from UCLA. And one of his quotes, he arrived at UCLA in 1948 as a junior and graduated and stayed to work for 50 years in the same place. And it was very difficult for him to live after such a long time at the age of 69, but still he did retire. What I like about that is, if I look at myself, I've been at this place for 15 years, and hopefully I can keep on going for another 35 years to reach 50 as well. So again, as if you're in a good environment, I think this, and you don't need to actually move. And I think UCLA for him was a great place to nurture his research and for him to really have a global impact in transplantation. But I'm very happy that after retirement, he said he tried to work from home, but it was impossible, and he established a Terasaki Foundation. And the good thing is, like he mentioned, that it was with a laboratory. So again, I guess the foundation that he wanted to establish still had to do some research, because another aspect of Dr. Terasaki also is a humanitarian aspect, and I think he created a foundation to really be able to help people from the different perspective. One thing that was really close to his heart, like I said, it was the Japanese American community, and always trying to help people from Japanese descent to achieve the best that they could. So again, he was really a philanthropic person, but still with this foundation, he still had a laboratory to start his research. And to me, what's good or important is this is actually the building where I started my research with him at the institute. And then again, in this foundation that he started, his first love of transplantation was really antibodies. And in 2003, he emitted this theory, the humoral theory of transplantation, that proposed for the first time that antibody were responsible for graft failure. So of course, he showed in 1969 that prior to transplantation, antibody were really bad for transplant. But after that, not many people believed that antibodies were really bad. So after also with the advancement of immunosuppression, really survival rates were really much better. And immunosuppression use is really focused on T-cell rejection or cellular mediated rejection, where antibodies don't really play a role. And because of all the improvement in survival in those patients, he always had to fight to say, look, guys, antibodies are important. And really for about 30 years, he had to really fight against the transplant community to show that, hey, guys, look at antibodies for your long-term survival. This is really what is important, because he always had in mind, the effort that we are doing also at the Institute is one transplant has to live forever in the recipient. And again, right now, the major hurdle in not achieving that one transplant for life is really the antibodies. And in 2003, he emitted that theory where nobody really believed in that. And what's great about this is that nowadays we know that that theory is actually true, and that antibodies are really one of the main culprits of a poor long-term outcome. But one thing amazing about this paper that was published in the American Journal of Transplantation is the editor was so amazed by how he showcased that antibody were deleterious to the organ. They actually decided to put his picture on the cover of a journal, which is extremely rare, and most of the time people don't just put the picture of the first author of the paper on the journal. So this really to showcase the impact of that publication for the transplant community. And here, so just a little bit of data, because we're still, doing some science. And here, really, those are the three main figures that he showed in that paper, really to demonstrate that with the presence of what he called cytotoxin, but those are cytotoxic antibodies, really the survival of a transplant recipient really gets lowered. And this, again, this was in 2003, and nowadays, this same observation are confirmed with newer cohorts of patients. And really, so this findings, or his interest in the humoral theory of transplantation, really made him develop this last, I guess, innovation was the single antigen bead assay, which allowed us to detect HLA antibodies in transplant recipients. Why it was a great assay is because we were able to multiplex, so test multiple HLA specificities in one run. So again, knowing that HLA is the most polymorphic gene in the human genome, you can create antibodies against many different HLA. And again, before they used to use cells of the donors to detect those antibodies, but again, in the post-transplant world, when you get an organ from a deceased donor, then you don't have access to cells from that donor anymore. So you still need a way to test for HLA antibodies, and this assay is really what allowed us to do so, doing post-transplant monitoring of HLA antibodies. So here again, to showcase that from his ideas that antibodies were bad for transplantation, he actually developed a new assay that helped him also demonstrate that his theory actually held true in patient. And again, with this assay, he was able to do post-transplant monitoring of those patients. So then the next step, I think in his life, which he was very proud of, is the Terasaki Life Science Building. So again, the Terasaki, our poor Terasaki and his family are philanthropic people, and really they always want to give back to the community. And I think Dr. Terasaki made this gift to UCLA, where they build a building, the Terasaki Life Science Building. And what's interesting, or what's to me what's great, is that at that time in 2010 is when I graduated from UCLA. So actually, I saw this building being built, but at first I didn't really know who, why, and the story behind it. But then in 2010, this is when I graduated from UCLA, and when I joined the Terasaki Institute at that time, and then finally understood why I had so much trouble parking at the school, because there was so much work done over there. And so here I want to talk now, I guess, a little bit about myself and what Dr. Terasaki did that I guess changed my life and my career path. Is really I was hired first as a laboratory technician, and again work in the lab and just doing experiment, analyzing results. But eventually my curiosity pushed me to start having hypothesis and thinking about writing papers. And interesting enough is I went to Dr. Terasaki. And I told him, like, hey, you know what, we should write this paper. And he told me, he's like, well, show me that you can. And I took the opportunity to do it, and then try to write papers to again try to help patients in the long run. But by doing this, I think one thing that I learned from him is again, doesn't matter what label or title you have, if you have the drive to do something, then you probably can achieve it. After, of course, you need to have a good mentor, somebody that stimulates you, and so on. But still, I think it's not the label that dictates what you can do. And I think, yeah, Dr. Terasaki believed that I could do it. And eventually I was able to publish papers. And from that, I was able to, get a PhD, but still working at the Institute. And eventually nowadays, becoming an assistant professor here, and I take a lot of pride in, getting my PhD here, because I'm kind of like the last, like, a scholar of Dr. Terasaki. And, one thing that I really want to continue is his legacy in the transplant field, but also how to have an impact on patient life down the line. And I think, he really taught me a lot, and it's really because of him that I'm here today, I think. So then, after that, in 2012, he received a great award, the UCLA Medal. And I think this also showed how he, had such an impact at UCLA, because this award is the, highest honor that the university gives. And again, showing that, Dr. Terasaki, impacted the life of, so many people, and especially Bruins. And I think also, because I was also a UCLA Bruin, helped me get the job out here, so he also liked the community of UCLA. So I think that's why also UCLA recognized him for that. So then after, also in 2014, I mean, this is the Terasaki Festschrift. So why I wanted to mention this thing, because I think, the whole institute came together to organize this event for Dr. T, which brought along all the transplant pioneer that were still alive, of course, at that time. And I think, you know, what I've witnessed there is what was his impact really in the scientific community, that he was able to move all the big players in the field to come and participate to this really great conference that we did here in LA. And also, you know, another part of Dr. Terasaki that, you know, didn't mention too much. And of course, I don't have enough time in 1 hour to talk about everything that he did. But one thing that he really enjoyed was, you know, the fun and shows and like music performances and things like that. And so this is a picture with many of us in the lab that are also here in the audience that, you know, we performed a little dance called the multi-night dance, which, you know, was trying to reflect, so a saying in Japanese where, like, you know, you have to really be careful with the resources that you have and not waste any resources. And I think we always had that state of mind in the lab to really never waste resources and being able to, you know, get the most out of what we had and really trying to have the most impact. And I think, you know, the Motana, you know, could be also seen as the, you know, when he developed the microcytotoxicity test by using only 1 microliter of sample, you know, again, he didn't want to be wasteful of really important material, but which was the thing that was great is really this put all of the people at the institute together, practice on dance, and eventually perform in front of all the people. So this was really a great moment, really also showcasing that, you know, working with him was really like being in a family. So then after coming to the end, you know, in 2016, fortunately, Dr. Terasaki passed away. But one thing that I want to mention is 2 weeks before his passing, he was still coming to the lab every day almost. And I think, you know, this shows a dedication and passion for the research and really wanting to continue stimulating the next generation of researcher. And again, this is one, you know, what we all had to write to in appreciation for what he did for us. But now for the good stuff is nowadays, you know, because of his vision, he was able to, you know, create this foundation, which, you know, what he said is that he hopes that this institute will continue to significantly support research for many years after he's gone. And I think we're still, you know, achieving that here. And again, you know, we're trying to still, you know, perform what he's tried to do before is the personalized medicine. So now we have a little bit more technologies available to us to do this type of research. And again, I think we're still trying to follow his guideline and vision on, you know, how we can have a real world impact and how to translate that technology into better patient care and quality of life. And so with that, thank you very much for commemorating his birthday today. And yeah, thank you. Well, yeah. Mhm.
Okay.
The beginning, you mix serum with the lymphocytes, then you mix serum with the antibodies. I'm a little bit confused. Can you explain a little more?
Well, so okay, so really the test that he did was a test to detect antibodies. And so again, he add antibodies to cells and see if it binds or if the antibody kills the cells. So here, what we have to remember is that in the discovery of HLA, we didn't know that HLA existed. So how the HLA was discovered was using the blood of multiparous women, so with multiple pregnancies. And pregnancy, we know that is a event that the sensitized mothers towards the paternal HLA. And so by seeing that, so they took those sera, tested against different cells and realized that there are different patterns of reactivities. And so then looking at different families, also seeing that like some reactivity then were from the mother or from the father. So the original idea was really more to see how antibodies bounded to cells. And by describing those patterns of reactivity, we could describe HLA molecules. So again, seeing how things are shared. And so by doing that is to type to really see who has which HLA. And then he leveraged that to say, hey, here I can test to see whether a transplant recipient has antibody against a specific donor. So when you do the typing, you're not using donor cells, you're using cells with known patterns of reactivity towards a specific serum. And so by analyzing those different patterns of reactivity, you can say, okay, this person is most likely to have this type of HLA or this type of HLA. But then again, because he was detecting antibody, he was able to also do that in transplant patient where there the setting is using donor cells and recipient sera to see if the recipient had any immune reaction towards the donor.
Thanks.
Great, again, Vadim, thank you for putting this together. I think it means a lot. I have a comment and then a question. For the comment, just for everyone here, right? I think what really struck a chord is when you talk about Dr. Terasaki actually, was rejected, right? A postdoc lab, and the first lab did it 1 year, and these are very famous people. I think just for everyone, I think it's very important when you do science. I think the persistence actually is the most important thing, right? I've advised so many students. I don't see a strong correlation. Someone came across as like super, like a genius, versus the final success. It's really the persistence and grit is always the best prediction because nothing worked from the first try. And if a great idea, if no one says it's a bad idea, it's probably not a great idea, right? Any great idea, you're taking a risk, and there are going to be a lot of pundits that that's a stupid idea, and that's usually how great idea come about. So I just want to kind of say that, right? Talking about, and even me too, right? Most of my grants get rejected, right? Most of my papers, and I think we all go through that. And I think as a trainees, just don't be discouraged. If you have a great idea, ultimately it's going to come out, but it's not going to come out, first paper. So you have to be really persistent on that, and eventually for the impact. Quick questions, yeah. Just anecdotal, right? In many cases, when I was hosting up, the chief editor from Science and other places, Terasaki, they say, oh, that's a Terasaki assay I learned. Is that the same Terasaki? And I was like, yes. I didn't have that history, but is that assay usually referred to the very first, the micro droplet toxin assay, or is it also the HLA,
So the Terasaki assay, I guess, yes, the microcytotoxicity test.
Cytotoxicity assay.
Yeah. After again, it's like in transplantation, his name always comes back everywhere. So for example, with HLA antibodies, there is the so-called Terasaki epitopes on HLA molecules and so on. So again, even at WTC last August, his name, is still quite alive in those meetings still. So, yeah.
Okay. Thank you. Thank you, Vadim. Thank you, everyone.
Thank you. Thank you. Thank you.